Dear students, today let us discuss about enzyme nomenclature and classification. Enzymes are biological polymers that catalyzes all biological reactions. Enzymes are large molecules that increase the rate of chemical reactions without themselves undergoing any change. In most cases, enzymes are proteins. The key aspect of an enzyme is that it reduces the activation energy required for a reaction. In doing so, it speeds up the reaction. Enzymes are vital in biological processes. Without enzymes, many chemical reactions in the cells which make the life possible would probably not even occur. So, we need to know how enzymes are named and the convention used for enzyme nomenclature. Many scientists try to assign enzymes with a specific name based on different criteria. However, they fail until the year 1955, where Enzyme Commission number, which is numerical classification scheme, was adopted. Background: the enzyme nomenclature scheme was developed starting in 1955 when the International Congress of Biochemistry in Brussels, the capital of Belgium, set up an enzyme commission. The first version was published in the year 1961. The sixth edition, published by the International Union of Biochemistry and Molecular Biology (IUBMB) in 1992, contains 3,196 different enzymes. Subsequent supplements have been published electronically at the website of the Nomenclature Committee of the International Union of Biochemistry and Molecular Biology. The IUBMB is an international non-governmental organization concerned with biochemistry and molecular biology. So far, IUBMB has 77 member countries. IUBMB organizes a triennial Congress of Biochemistry and Molecular Biology and sponsors more frequent conferences, symposia, educational activities, and lectures. It publishes standards on biochemical nomenclature, including enzyme nomenclature, in some cases jointly with the International Union of Pure and Applied Chemistry. Nature of enzyme reactions. The cells in our body are chemical factories. Most of the substances needed by our body are synthesized within our cells, which means that hundreds of chemical reactions takes place in our cells every second of our life. We call molecules that participate in a reaction moderated by an enzyme as reactant or substrate. We will use both terms interchangeably. The end product of the reaction are called products. During a reaction, the enzyme binds to one or more reactants, and it is the binding of the enzyme to the reactant that lowers the activation energy. It is truly fascinating that this process occurs in nature, allowing life to exist. We call the site to which a substrate binds to the enzyme the active site. The substrate can become bound to an enzyme with the following interactions, such as hydrogen bonding, van der Waals interactions, electrostatic attractions, etc. Enzymes are commonly given names derived from the reaction they catalyze and the compound or types of compounds on which they act. For example, lactate dehydrogenase, which speeds up the removal of hydrogen from lactate, an oxidation reaction. Similarly, acid phosphatase catalyzes the hydrolysis of phosphate ester bond under acidic conditions. As can be seen from this example, the name of most enzymes end with a suffix ase that is appended to the name of the substrate associated with the enzyme. Another example is lactase, breaking down of the sugar lactose into galactose and glucose. Hexokinase, which convert any hexose into hexose monophosphate. Similarly, alcohol dehydrogenase, which convert ethanol into acetaldehyde. While not all enzymes are named using this convention, some enzymes, however, have older names, which were assigned before their actions were clearly understood. Among these are pepsin, trypsin, and chymotrypsin, which are all digestive enzymes. General information on enzyme nomenclature. Before an enzyme can be placed into a particular list, direct experimental evidence is required that the proposed enzyme actually catalyzes the reaction claimed. Close sequence similarity is not sufficient without evidence for the reaction catalyzed, because only a small change in sequence is sufficient to change the whole activity or specificity of an enzyme. Furthermore, because classification is also based solely on the reaction catalyzed, there are cases where proteins of very different sequences catalyze the same reaction. All enzymes are named systematically according to a classification scheme designed by Enzyme Commission, that is E.Sc, of the International Union of Pure and Applied Chemistry, IUPAC, that is based on the type of reactions they catalyze. Each enzyme type has a specific 4-integer E.Sc number. This is a complex system, but it is unambiguous that eliminates confusion between enzymes catalyzing similar but not identical reactions. In practice, many enzymes are known by a common name that is derived from the name of its principal specific reactant with a suffix ASE. Some common names do not even have ASC appended, but these tend to be an enzyme that were studied in the early days before the systematic classification of this enzyme had been assigned. The Enzyme Commission number, E.Sc number. E.Sc number is the numerical classification scheme for enzymes based on the chemical reactions they catalyze. As a system of enzyme nomenclature, every E.Sc number is associated with a recommended name for the respective enzyme. Strictly speaking, E.Sc number do not specify enzymes, but enzyme catalyzed reactions. If different enzymes, for instance from different organisms, catalyzes the same reaction as in the case of aldolase, then they receive the same E.Sc number. In addition, through conversion evolution, completely different protein fold can catalyze an identical reaction and therefore would be assigned an identical E.Sc number. These are called non-homologous isofunctional enzymes or NISE. Format of number. Every enzyme code consists of the letter E.Sc followed by 4 numbers separated by periods. Those numbers represent a progressively finer classification of the enzyme. In one example, the tripeptide aminopeptidase have the code EC34114, whose component indicates the following groups of enzymes. EC3 enzymes are hydrolase, that is, enzymes that use water to break up some other molecule. EC34 are hydrolase that act on peptide bonds. EC3411 are those hydrolase that clip off the amino-terminal amino acid from a polypeptide. EC34114 are those that clip off the amino-terminal end from a tripeptide. Enzymes are classified by reaction type and mechanisms. A system of enzyme nomenclature that is comprehensive, consistent, and at the same time easy to use has been proved elusive. The common names for most enzymes derive from their most distinctive characteristic, that is, their ability to catalyze a specific chemical reaction. In general, an enzyme's name consists of a term that identifies the type of reaction catalyzed followed by the suffix ASE. For example, dehydrogenases such as lactate dehydrogenases, which remove hydrogen atoms, proteases which hydrolyzes proteins, and isomerases such as phosphohexose isomerase catalyzing rearrangement or configuration. One or more modifiers usually precede these names. While many modifiers name the specific substrate involved, such as xanthine oxidase, others identify the source of the enzyme, pancreatic ribonucleases. in some cases specify its mode of regulation, hormone-sensitive lipase. When it was discovered, multiple forms of some enzymes were existed. Alphanumeric designators were added to distinguish between them. For example, RNA polymerase 1, 2, and 3. To address the ambiguity and the confusion arising from this inconsistency in nomenclature and continuing discovery of new enzymes, the International Union of Biochemists (IUB) developed a complex but unambiguous system of enzyme nomenclature. In the IUB system, each enzyme has a unique name and code number that reflect the type of reaction catalyzed and the substrate involved. Although common names for many enzymes remain in use, all enzymes are now classified and formally named according to the reaction they catalyze. 6 classes of reactions are recognized, such as oxidation-reduction reaction, functional group transfer reaction, hydrolysis reaction, isomerization reaction, etc. Within each class are subclasses, and under each subclass are sub-subclasses, within which individual enzymes are listed. Classes, subclasses, sub-subclasses, and individual entries are numbered so that a series of 4 numeric numbers serves to specify a particular enzyme. For example, the enzyme commonly called hexokinase is designated ATP dehexo-6-phosphotransferase or EC2711. This identifies hexokinase as a member of class 2 transferases. Subclass 7, that is the transfer of a phosphoryl group. Subclasses 1 means alcohol is phosphoryl acceptor. And finally, the term hexo-6 indicates that alcohol phosphorylated is at the carbon 6 of hexose. EC2711 having a common name of hexokinase, which catalyzes the reaction of ATP plus dehexose giving ADP plus dehexose 6-phosphate, which has a systematic name of ATP dehexo 6-phosphotransferase. Similarly, EC2712 having a common name of glucokinase, catalyzing a reaction ATP plus D-glucose giving ADP plus D-glucose 6-phosphate, which have a systematic name of ATP D-glucose 6-phosphotransferase. To illustrate, consider the enzyme that catalyzes this reaction. ATP plus D-glucose, which gave ADP plus D-glucose 6-phosphate. A phosphate group is transferred from ATP to C6OH group of glucose. So the enzyme is transferase, that is class 2. Subclass 7 transferase is enzymes transferring phosphorus containing groups, and subclass 1 covers those phosphotransferase with an alcohol group as an acceptor. Entry 2 in this subclass indicates glucose 6, and thus it is ATP D-glucose 6-phosphotransferase, and its classification number is EC2712. For example, entry 1 in the same subclass is EC2711, and it is for ATP dehexose 6-phosphotransferase, an ATP-dependent enzyme that transfer a phosphate to the 6-OH of hexose. That is, it is non-specific regarding its hexose acceptor. The glucose-specific enzyme EC2712 is called glucokinase, and the non-specific EC2711 is known as hexokinase. Thus, kinase is the general term of enzymes that are ATP-dependent phosphotransferases. In top-level EC numbers, enzymes are classified into 6 groups: oxidoreductase transferase, hydrolase, lyase, isomerase, ligase. Group EC1 is the oxidoreductase reaction catalyzed oxidation reduction reactions, that is transfer of hydrogen and oxygen atoms or electrons from one substances to another. Typical reactions here is AIs plus B giving A plus BIs, Here B is undergoing reduction. A plus O giving AO. Here A is undergoing oxidation. Examples are dehydrogenases, oxidase. Group EC2 transferase. Reaction catalyzed is the transfer of a functional group from one substrate to another. The group may be methyl group, acyl group, amino group, or phosphate group. Typical reactions here is AB plus C giving A plus B.Sc, where B group is transferred from A to C. Examples are transaminase kinase. Group EC3 hydrolase. Reaction catalyzed is the formation of two products from a substrate by hydrolysis of water. Typical example here is AB plus water, it gives AOH plus BH. Here the substrate A is undergoing hydrolysis. Examples lipase, amylase, peptidase. Group EC4 lyase. Reaction catalyzed is non-hydrolytic addition or removal of groups from substrate. Here carbon-carbon, carbon-nitrogen, carbon-oxygen or carbon-sulfur bonds may be clipped. Typical reactions here is RCO-COOH giving RCOH plus carbon dioxide. Here carbon dioxide is removed from the substrate by cleaving CC bond. Examples decarboxylase. Next, group EC5 isomerase. Reaction catalyzed is the intermolecular rearrangement, that is isomerization or changes within a single molecule. Reaction catalyzed, say for example ABC giving BCA. Here rearrangement of atoms within the same molecules takes place. Examples are isomerase mutase. And finally, group EC6 ligase. Reaction catalyzed is joining of two molecules by synthesis of new bonds such as carbon-oxygen, carbon-sulfur, carbon-nitrogen or carbon-carbon with simultaneous hydrolysis of ATP. Typical reaction is X plus Y plus ATP giving XY plus ADP plus inorganic phosphate. Here formation of new bond amongst X and Y with the energy coming from ATP hydrolysis. Example is synthetase. Special examples. Sometimes there are cases where we need to name an enzyme based on the overall reaction catalyzed by a group of enzymes. Here, the word system should be included in the name. For example, the system catalyzing the oxidation of succinate by molecular oxygen, consisting of succinate dehydrogenase, cytochrome oxidase, and several other intermediate carriers. In this case, it should not simply name as succinate oxidase, but it may be called the succinate oxidase system. Other examples of system consisting of several structurally and functionally linked enzymes and cofactors are the pyruvate dehydrogenase system, 2-oxoglutarate dehydrogenase system, and the fatty acid synthase system. Conclusion. So in conclusion, there are several types of enzyme catalyzing different types of biological reactions, and thus we need to classify them and understand the proper nomenclature for every single types of enzyme. So whenever a new enzyme comes up, it would be taken care by the nomenclature committee of the International Union of Biochemistry and Molecular Biology. For naming an enzyme, a systematic non-ambiguous enzyme commission number having a 4 integer is used. These 4 numeric numbers signify for class, subclass, sub-subclass and individual entries for a particular enzyme. Thank you.